Hello and welcome to the last part of the course, MOL200. For the next 3 lectures, we're going to discuss chapter 23. There's quite a lot in it, but we'll try and go through it slowly so that you get the best impression of the content. So basically what we want to discuss is how the chemical energy from food is converted and distributed throughout the body. So we have a set of learning goals. We want to know how the different tissues divide up the work. What's their function and how they work together. And we will look at then the metabolism of sugars, amino acids, and lipids, things we've covered already in the different tissues. In part, we've covered this already, so we'll go quickly through those parts. And perhaps more importantly is to understand how the different organs communicate with each other and pass metabolites between each other. So two important regulators of this are insulin and glucagon that we have met already again, and we'll just try and get a sort of an overall picture of how these things are working before we move further in the next part into discussing the effects of fasting and starvation on metabolism. Okay, so here we see most of the important organs in the body taken out here, displayed. They don't show us the kidneys. We will be mentioning the kidneys. We have mentioned it before. Their role is to export glucose under starvation conditions via gluconeogenesis, and then of course to remove waste, urea, that builds up in the body. We will meet the pancreas, which secretes insulin and glucagon. We will talk about the liver, its many roles. It processes fats, sugars, proteins from the diet, and then it also synthesizes and distributes lipids, ketone bodies, and sugars again, glucose, for other tissues. It's an important function of it. So it's this altruistic or unselfish tissue that functions to benefit the other tissues. And it also converts nitrogen to urea. We will briefly mention the intestines and the blood system that are of course important for absorbing food and for distributing food to different tissues. Muscle we've talked about already and we'll mention again. So it uses ATP generated aerobically or anaerobically to do mechanical work. Adipose tissue we will discuss. It synthesizes, stores, and mobilizes triacylglycerols. That is the white adipose tissue, and we will mention briefly brown adipose tissue, which carries out thermogenesis, so the generation of heat. And the lymph system, which carries lipids from intestine to liver, you've probably already covered. And then the heart, just briefly, we will mention that it uses ATP generated aerobically, and its function is to pump blood. The processes that we will discuss going on in these different tissues are processes we have covered already by this stage, and we will just refer to them, right? So carbohydrate metabolism, or catabolism and anabolism, so building up and breaking down of sugars, amino acid metabolism, fat metabolism, and these are pathways that you have already covered, and we will just refer to them and try and give you this impression of how the organs are working together through these pathways. So I showed this slide in the orientation meeting, and the next slide, to show you in a way that these pathways that we have covered as individual blocks of information, they act in concert together, right, under different conditions. So here we have the fed conditions, and then we have the starvation conditions. So in fed, these pathways that we learned about are on, glycolysis is on, glycogen metabolism, you're making glycogen, fatty acid synthesis is on, etc., etc. Citric acid cycle, these are on during the fed conditions. But during starvation conditions, we have a switch, but it's a coordinated switch. We have gluconeogenesis and we have the urea cycle being turned on, we have increased beta oxidation. And these are adaptations in the cell to ensure that the organism survives. So we have sort of changes in these pathways at a cellular level, and then we have changes in the function of organs at an organismal level as well, that we have to sort of reconcile and ourselves and build a picture of how these things work under different conditions. And in the third talk, so 3 talks, in the third talk I will discuss a little bit disease states that are relevant when it comes to metabolism. So first, start with the basics, the liver, the body's distribution center, right? So the liver, the cells are called hepatocytes. They are metabolically flexible, so they adjust to nutrients and needs of the body in response to hormonal signals, as we discussed. They have a high enzyme turnover, 5 to 10 times the normal going on in other cells and other tissues, and they're specialized to export nutrients. The overall role of the liver is to smooth out fluctuations in metabolism or changes in energy levels or nutrients in the bloodstream. And additionally, they function as a storage of iron and vitamin A, and are involved in detoxification of poisons that come into the bloodstream. The liver serves as an altruistic metabolic hub for the body. So it coordinates the exchange of metabolites between major tissues and organs. So this picture gives us a sort of a snapshot of what can go on. And if we look then at the liver, it's obviously very central. We see here we have glucose. Glucose can have many fates in the liver. It can be converted to glycogen as a store, or it can be exported from the liver, primarily as a source of glucose to the brain, but also secondary role would be distributing glucose to adipose tissue and muscle. It can also break down glucose to pyruvate via glycolysis and then to acetyl-CoA, and this acetyl-CoA can be used to generate fatty acids, and these can then be exported to the store, the adipose tissue. And the primary function of the adipose tissue is to store fatty acids, right? Or they can be supplied to active tissues requiring energy, like, for example, muscles. And then here we have the intestine. We get our food comes in, right? So the glucose, amino acids, and fat, and then they're sort of delivered via different mechanisms. The fats via the lymphatic system and can be taken to the liver or even distributed to metabolically active tissues like the muscles. Amino acids through the blood together with glucose and enter into the liver. So the liver is a central hub for receiving and distributing metabolites. Now, I include this figure. It's not in Leninger, it's in another book. I think it gives a helpful overview. So first of all, it also includes the kidneys and the heart. But if you look at this core here, it's very similar to the last slide. So we won't go through the whole thing again, just point out some extra things that are on here. So the liver, it's communicating with the muscle in different ways. So for example, a supply of triacylglycerol, which can be released and fatty acids delivered to the muscle for use in beta oxidation. It also, we have the buildup of pyruvate upon glycolysis in the muscle when the muscle is active. And the muscle will convert pyruvate to lactate under anaerobic conditions. And this will be a problem. You get a buildup of lactate in the muscle, and the lactate is actually transported back to the liver where it can be converted to pyruvate and then further via gluconeogenesis to glucose and redistributed back to the muscle. This is referred to as the Cori cycle. We'll meet this a little bit later on. And it's a energetically expensive way to keep the muscle supplied with glucose, as we'll see. Additionally, we have conditions where amino acids can be used to generate pyruvate in the muscle, sticking with the muscle, and eventually ATP. And the breakdown of amino acids typically leads to a buildup in urea. And one of the ways to get rid of urea is to transport alanine to the liver, where it can be converted to pyruvate, and the urea, the ammonia, can be removed from the amino acids and sent to the kidney. And then the alanine converted pyruvate can be again converted to glucose in the liver and sent back to the tissue. And this is referred to as the alanine glucose cycle, and we'll have a look at that a little bit later on as well. So there's these rounds of these cycles of sending metabolites, using them up in the muscle and sending the waste almost to the liver, for the liver to use energy to make them useful again for the muscle and then sending them back. And we mentioned the urea would be excreted by the kidney, so it needs to be delivered there. But also amino acids like the alanine can be delivered to the kidneys for conversion back to glucose and release out. And then all of these gluconeogenic tissues like liver and kidneys work to keep the brain well fed with glucose. I think that's the most important points on this slide. We'll meet the slide again where relevant to try and help us keep an overview of what's going on. So when it comes to the use of sugars in the liver, we have met this, but not as completely as it is presented here. So we will go through it. So we have glucose in the liver, or glucose in the blood. We can even start, right? So the liver cells, the hepatocytes, they express the GLUT2 transporter, which allows for the diffusion of glucose down a concentration gradient. So if you have more glucose in the blood than in the hepatocyte, glucose will enter the hepatocyte. If there's more glucose in the hepatocyte than in the blood, it can go out and back into the blood. So that's one of the specialized functions of the liver, that it can allow glucose to escape its cells, again being altruistic, right? So if you have glucose coming in, typically it will be converted to glycogen and used as a liver store of glycogen, which can then be broken down under starvation or semi-starvation conditions to glucose again. The glucose 6-phosphate, so when you break down glycogen, you get glucose 6-phosphate, and this had various fates that we discussed already. It can enter into glycolysis to generate pyruvate, and that pyruvate can be converted to acetyl-CoA, enter the citric acid cycle to generate ATP. But liver cells don't do that so much with glucose, right? So actually fatty acids are the preferred source of energy in the liver for OXPHOS. And this is due to the expression of a transcription factor called PPA or alpha, peroxisome proliferating something. PPA or alpha expression, which drives expression of genes involved in beta oxidation, causing increased beta oxidation in hepatocytes. And we will meet this a little bit more in detail after. Not much, but a little bit more. Try to make things a bit more clear. So what happens if the glucose that's converted to acetyl-CoA is not taken into the citric acid cycle? What happens to it? Well, it can be used for other things. So the liver is a major site of cholesterol synthesis in the body for export to other cells. Also, the liver then can convert this acetyl-CoA to fatty acids and then pack them as triacyl glycerols and export them in the bloodstream, or use it for its own cell membranes if it wanted to. Then the last way in which glucose 6-phosphate can be used is via the pentose phosphate pathway to generate nucleotides. Again, if the liver cells want to make new cells, they will need new nucleotides, right? And this was also an important way to regenerate NADPH in the liver. There were some special features that we have discussed already, like the liver had expression of hexokinase 4 or glucokinase, which had a high Km, so it didn't recognize glucose that well. It didn't convert low levels of glucose to glucose 6-phosphate, but high levels it did. And this was, in fact, an important step to trap the glucose in the liver cell, in the hepatocytes, right, because of this GLUT2 transporter. So the major function when it comes to sugars in the liver is to keep blood glucose balanced at a sufficiently high level, a concentration of about 4.5 millimolar or 80 milligrams per deciliter. If blood glucose drops to, for example, 3.5 millimolar, you will develop feelings of lightheadedness, maybe some mild confusion. If it drops again below 2.5 millimolar, this will lead to serious effects, including eventually death. And we'll have a look at this in the second lecture of these three. Now let us turn to the fate of amino acids in the liver. So amino acids can, of course, be incorporated into liver proteins, or they can be incorporated into proteins that are destined for secretion into the bloodstream. Additionally, without being converted into proteins or incorporated into proteins, the amino acids can be released into the blood as a source of fuel for other tissues. And then amino acids can be converted to nucleotides, some hormones, and porphyrins, which are molecules like heme, oxygen binding heme. So amino acids can be metabolized, source of energy within the liver, broken down to pyruvate, and this will come at the cost of producing ammonia, which has to be converted to urea via the urea cycle, and then they can be secreted and excreted from the kidney. Then the liver has this specialized gluconeogenesis function, where pyruvate can be converted to glucose and exported. So amino acids can generate glucose in the liver for export in the blood. Amino acids can be broken down to acetyl-CoA, enter into the citric acid cycle to provide a source of ATP. And some amino acids, you probably heard about this when you were listening to Øyvind talking about amino acid metabolism. Some amino acids can be used for gluconeogenesis to provide glucose via the citric acid cycle. And again, that glucose can be exported to the bloodstream and delivered to other tissues. Sorry, I was blocking down there. there wasn't that much to see down here. It was just oxidative phosphorylation. So the final point on this slide was the amino acid function in the liver, and sometimes the amino acids are coming from the muscle. So if the muscle is, we can look from this part here over here. It's a bit more clear, I think. Muscle proteins broken down to give energy ends up with a buildup of ammonia, and then there's an enzyme, alanine aminotransferase, that takes pyruvate and glutamate and the ammonia and makes alpha-ketoglutarate and alanine. Then the alanine is exported via the blood to the liver, where we have the almost opposite reaction, where the alanine and the alpha-ketoglutarate via, again, the enzyme alanine aminotransferase give rise to pyruvate and glutamate and ammonia. And the ammonia then can be taken out by the urea cycle, exported to the kidneys for excretion. And the pyruvate then, via the input of 6-ATP and gluconeogenesis, is made back to glucose in the liver and exported via the blood back to the muscle to give more glucose to the muscle. So again, this is an example of the muscle being a selfish tissue and the liver being an altruistic tissue. And the muscle can't perform gluconeogenesis, right? But it needs glucose. So under certain conditions, starvation, when proteins are being broken down for a source of energy, then this is an important way to keep the muscle. And, it doesn't have to go back to the muscle. It can go to the brain instead, right? The glucose doesn't know where it's going to go. It will go to the tissue that needs it. So when it comes to the fate of lipids or fatty acids in the liver, we have fatty acids here. We see that one of the routes they can be utilized in is making liver lipids, for example, to make the plasma membrane. Otherwise, they can undergo beta oxidation to give us acetyl-CoA for the citric acid cycle and oxidative phosphorylation and generation of ATP. And in the liver, as I said, fatty acids is the preferred source of metabolite to power oxidative phosphorylation and make ATP. And this is because of the high expression of a transcription factor, PPA or alpha, which increases beta oxidation in this tissue. So that again, so the glucose that's taken into the liver can be used in other ways for other tissues, right? Acetyl-CoA that enters from fatty acids can be converted again to cholesterol. The major site of synthesis of cholesterol in the body is the liver, and these are exported. The cholesterol can be converted to steroid hormones and exported in the blood, or the cholesterol can be exported in the blood. Also, in the gallbladder of the liver, cholesterol can be converted to bile salts, which are important in the digestive process. Over here, fatty acids in a hepatocyte, plasma lipoproteins. Right, so these vesicles that travel around delivering a fat content to cells. And then, some free fatty acids in the blood, not completely free, but they're bound to albumin and delivered to tissues so that they can be used for those tissues metabolism, like the muscle burning fatty acids can be delivered this way. And then I forgot to mention, of course, very importantly, under starvation conditions, fatty acid, beta oxidation, and acetyl-CoA can lead to the generation of ketone bodies, acetoacetate and beta hydroxybutyrate, which can be exported from the liver to the blood and used to fuel tissues, including the brain, the glucose-dependent brain. But fatty acids broken down to acetyl-CoA cannot contribute to gluconeogenesis, so they can't make glucose out of fatty acids. So now we come to this somewhat messy slide. I'll get out of the way again just so you can see this part, even though this part is not on the test, let's say. So up here, some metabolic integration by PPARs. So there's 3 at least PPARs: alpha, gamma, delta, and these are nuclear receptors that are activated by fatty acids, free fatty acids. So fatty acids act as a ligand to bind to these and trigger transcriptional changes in the nucleus. 3 PPA or isoforms regulate lipid and glucose homeostasis through their coordinated effects on gene expression in the liver, muscle, and adipose tissue. So PPA or alpha and delta, and its closely related isoform beta, not shown here, regulate lipid utilization. So fatty acid oxidation, response to starvation in the liver, and fatty acid oxidation and thermogenesis in adipose tissue. So this is a special type of use of the fatty acids that we will see when we look a bit more closely at adipose tissues. PPA or gamma regulates lipid storage and insulin sensitivity of various tissues. We don't need to know more than this. So alpha is important in the liver. It promotes fatty acid oxidation. It's also, we have a lot of fatty acid oxidation in the muscles as well, right? And then this thermogenesis is something that we will have a look at almost right now. So adipose tissue, there are 2 types: white adipose tissue and brown adipose tissue. And the functions and the physiology and their appearance is different. So when it comes to function, white adipose tissue is storing energy, whereas brown adipose tissue is expending energy and heat production. So this non-shivering thermogenesis. So shivering, when you're cold and you're shaking, keep warm, that's your muscles are, contracting, and you will generate some heat by that work that's done. And this is non-shivering thermogenesis, where there's actually this distribution of fat in different parts of the body. In brown adipose tissue, you see in the baby, it's up on the shoulders and on the back, and in the adults, it's around the spine and shoulders and some of the vertebrae and some of the internal organs. And it will actually, when you're cold, it will start using energy to heat up. And if you look on a heat camera, you see the person here at 27 and you shift them to cold 18. On the heat camera, you see the buildup or the release of heat from this brown adipose tissue, and this lets us know where it's distributed in the body. So this is a way that the body can protect vital organs and presumably the spinal column's function by keeping it warm at the expense of burning energy, keeping your body functioning. So they look a little bit different. The brown adipose tissue has many small lipid droplets, many mitochondria, and then they express a protein called thermogenin, or uncoupling protein 1, UCP1, to decouple oxidative phosphorylation. And then from beta oxidation, we get heat being made instead of ATP. So if we look here, we have the oxidative phosphorylation chain, and here we have the protein thermogenin, and it decouples the protein gradient by allowing hydrogen ions to flood into the mitochondrial inner space. The membrane potential will be disrupted, and instead we get a release of heat instead of ATP generation. And this is actually pKa activated. I think the expression of the UCP gene is under the control of some of the PPARs, the gamma and alpha it says here, and maybe the delta. I mean, it would make sense that it was the delta because that's what it says in the slide. But when I look online, I get some different things. We don't need to know so much. Let's go with what the book says. The simplest is the delta drives the expression of UCP1 in brown adipose tissue. and then these are able to be to decouple the proton gradient when pKa activates. And what would activate pKa? Well, it could be adrenaline, for example, if you were cold. No. The white adipose tissues, they are the major lipid store in the body, typically 15 to 20% of body weight, or up to 35% in the extreme cases of obesity, are due to white adipose tissue. When it comes to the storage of lipids, so about 65% of the cell is taken up by one large lipid droplet, compared to the brown adipose tissue where we had several small. Here we have just one large store, and they are stored as triacylglycerols. These tissues are very metabolically active and respond quickly to hormones, and they have an interplay. So adipose tissue has an interplay with liver and muscle when it comes to releasing fat and delivering fat to the muscles, and unused fat delivered from adipose tissue to the muscles needs to go back to the liver to be packaged and sent back to the adipose tissue for storage. So that's that interplay. Just briefly, they have a high glycolytic metabolism. They use the citric acid cycle to oxidize pyruvate and fatty acids, and they carry out oxidative phosphorylation. And this is what I said here. If we look at fat mobilization from these white adipocytes, adipose tissue cells, we have the triacylglycerol cycle. So what's important here? We have triacyl converted to fatty acid. This is regulated by hormone-sensitive lipase, which will release the fatty acid from the glycerol backbone. This hormone-sensitive lipase is regulated hormonally by glucagon and epinephrine, activating it, driving the breakdown of triacylglycerol and release of fatty acids, and negatively regulated by insulin. So when you have a lot of blood glucose, insulin signaling will be high, will shut down the release of fatty acids because your body can now use glucose and store. Lipids are a better store of energy, right? Conversely, when glucose levels drop, you will have, of course, gluconeogenesis and breakdown of glycogen, but glucagon and epinephrine can signal to hormone-sensitive lipase to also start releasing the fatty acids. Okay, so now you have fatty acid in this cell. It can be exported as free fatty acid or bound to albumin and delivered to tissues. And these tissues should have expression of this lipoprotein lipase to allow it in. So the free fatty acid fuel for tissues. So on muscles, for example, heart, and on adipose tissue, we have this lipoprotein lipase that is needed to get the fatty acid back into the cell. Okay, so we have the free fatty acid. It can be delivered from the adipose tissue via the blood to tissues to fuel beta oxidation and give them energy. The unused fatty acid is taken up by the liver, converted back to triacylglycerol, and then secreted out. So when glucose again becomes plenty, let's say, the unused fatty acid is packaged into triacylglycerol again, delivered via very low density lipoproteins, VLDL, to the adipose tissue, and the adipose tissue expresses a protein called lipoprotein lipase, which again hydrolyzes the triacylglycerol to free fatty acids and glycerol, and then the free fatty acid is converted to triacylglycerol again and stored in the lipid droplet in the white adipose tissue. So that's the most important parts of that, I think. So what's another could be good to keep in mind, approximately 75% of the fatty acids are re-esterified to triacylglycerols in the adipocytes. So this is a kind of a substrate cycle. So what does that mean? 25% will be used by fuel for tissues. The rest will be recycled back and not used. So kind of, again, you can think of this as not being the most energy efficient way to do things, but you are supplying fuel for potential needs, let's say like that. Okay. You have a margin, a safety margin of most of the fatty acids will not actually be used in that exercise, but they need to be, again, then stored, and this is the mechanism for that. When it comes to muscles, there are, the muscles are these cells that will fuse with each other and have many nuclei in the end, right? They're big, long cells or fibers. And you can have fast twitch or slow twitch muscle fibers, and the fast twitch are the ones you build up during sprint exercises, and the slow twitch are the ones you build up during marathon running. And basically, you have both in the muscles, but the proportion of fast or slow twitch changes dependent on the type of exercise you do. And the metabolism of both these types is slightly different. This just shows you, here we have a cross section through a muscle, showing us darker red and the lighter. So you have both of them there. Both can use free fatty acids, ketone bodies, and glucose. Here is not on the test, just a list of things for comparison, the different ways the muscles function. The fast twitch muscle fibers are, they undergo aerobic and anaerobic, whereas the slow twitch mainly undergo aerobic. The fast twitch are better in burning fat, and the slow twitch prefer glucose burning. So they have substrate specificity or substrate preference differences, let's say. And that's of course, some have more blood flow than others, right? That was one of the things that just pointed out. That's why you get the color. So the slow twitch muscle fibers will have more blood flow, right? Fast twitch are less red, they have less blood vessels. The heart is dependent on oxygen. It has to be kept oxygenated. It gets its energy under aerobic conditions at all times. This is a cross section of it. You see it's abundant mitochondria flanking the muscle fibers. So free fatty acids are the main fuel, but it can use glucose and ketone bodies in the worst case. These are all oxidized via citric acid cycle and oxidative phosphorylation. So this is why we need oxygen all the time. So just a few more words on the substrate preferences in the muscle. It depends on what the muscle is doing, right? So during bursts of heavy activity in the muscle, glycogen is broken down, and we discussed this in lecture 7. It's broken down to give lactate with the generation of 3 ATP. Then we have, again, under bursts of heavy activity, phosphocreatine can be used as a rapid source to replace ATP. So it takes ADP and converts it to ATP. Under conditions of light activity or rest, the muscle will use fatty acids, ketone bodies if they're available, or blood glucose and break them down completely and extract a lot of ATP. This is just to remind you, pyruvate to lactate differs a little bit from pyruvate to acetyl-CoA, in that here we can, when we make lactate, we can replenish NAD+, and this is important for glycolysis. And also then you get this is shown here. When you break down glycogen, you can skip ATP investment step to give us glucose 6-phosphate. We directly make glucose 6-phosphate almost. And So rather than consuming 2 ATP in the investment phase of glycolysis, we only consume 1, so therefore we can net 3 ATP instead of 2 via this or this route. So we save an ATP, which is important for the muscle under anaerobic conditions. This we have covered before. Just to remind you, the creatine kinase reaction takes phosphocreatine and ADP, and creatine kinase then converts it to ATP and creatine. And this reaction was concentration dependent, so when ATP levels are restored after exercise, it will shift in the opposite direction, and phosphocreatine can be built up again as a reservoir of rapidly available energy. Right, one of these high energy molecules we discussed early on. So one of the ways that muscle communicates with other tissues is via the Cori cycle. It communicates, I mean, it is dependent on other tissues like the liver. It's called the Cori cycle. So, after or even during exercise, you end up with heavy breathing, pulling in a lot of oxygen, and this is in an effort to replace the ATP that's used or to enable fatty acid oxidation. These are things you need oxygen for. This is the figure from the book, and this is one that explains it perhaps a little bit better, but you can see here. So glycogen stores in the muscle broken down to release ATP and lactate. Lactate is secreted in the blood, picked up by the liver, converted via the use of ATP to glucose, which is recycled back. So then you're making this ATP. You're going to need oxygen, more oxygen in the liver to do this, and you start sucking in the oxygen. Right? So even if it's anaerobic exercise on your muscle, you're going to need the oxygen in your liver. This is explained a little bit more here. Glucose glycolysis, pyruvate, get lactate dehydrogenase enzyme, lactate exported. And then we have the conversion of lactate to pyruvate, and from there gluconeogenesis at the expense of 6 ATP. So if we're breaking down from, let's be kind, we say breaking down from glycogen, we can get 3 ATP out, and then to get those 3 ATP out in the muscle, we have to spend 6 ATP in the liver to regenerate that glucose, let's say. So the liver is not prioritized in this way. Another important function of this lactate dehydrogenase shunt was the restoration or replenishing NAD, which is needed for glycolysis right up here. So in this way, that the muscle can continue to anaerobically break down glucose to pyruvate and extract this 2 ATP if it's recycled back. And then that will cost us 6 by this 6 ATP in the liver for 2 ATP in the muscle. So the muscle is prioritized in terms of energy expenditure compared to the liver. And then these two cycles that we talked about recently are shown here on this schematic that I showed you. That was not in the book, but we saw that so the lactate built up in the muscle could be converted back to glucose and cycled back via the Cori cycle. And in the case when amino acids are broken down, we get a buildup of ammonia that's coupled to alanine synthesis, and alanine is transported to the liver or to the kidney, where it can be converted back to glucose, and the ammonia can be removed. You get a buildup of urea excreted from the body. And then I just want to point out finally that the muscle can, under extended periods of use, so like endurance sports, the muscle can get energy out of fatty acids as well, right? So we have it's not storing them typically, we have the storage in the fat, we have the triacylglycerol cycle communicating with the liver, releasing via lipoprotein lipase, releasing triacylglycerols, and the liver can package these for delivery to the muscle. where the muscle then can break down fatty acids to make acyl-CoA and generate ATP. So if we were to think about the start of a run or a sprint that turns into a marathon, how would we use the different resources? How would our muscles use the different resources? Well, ATP that's already there would be used up first and very rapidly, right? And then we mentioned we have this muscle phosphocreatine that can convert phosphocreatine to ATP or ADP to ATP via the use of this phosphocreatine. And then of course we have these are very rapid responses, right? And then the breakdown of muscle glycogen is enough to keep us going for a while after that when these stores are getting low, in order to break down the muscle glycogen to keep the muscle ATP at a reasonable level. And then following the use of muscle glycogen, we can start mobilizing fatty acids. This process takes a bit longer to get going, right? And then the muscle can run on fatty acids as a source of energy. So this brings us to the penultimate tissue we'll discuss. We're coming close to a break here in this part of the lecture, at least. The brain. And the brain uses glucose as fuel in the neurons. The brain isn't only neurons, it's also glia, and they can actually oxidize fatty acids. But a large part of the brain is totally dependent on glucose. In fact, when the body is at rest, 20% of total oxygen usage is done by the brain. So the breaking down glucose and oxphos, 20% of the energy in the body at rest is done by the brain, is used by the brain, right? And the brain's, when you think about brain mass to body, it's only a few percent of the total body weight, but it's using 20% of the energy in the form of glucose and other things, but yeah, energy, let's say. So the brain has very little glycogen storage. So I think these astrocytes had some. So it's totally dependent on glucose supply, and too low glucose may induce serious damage. And the reason why we need so much energy in the brain is because it's composed completely of neurons, and neurons, in order to have a potential to signal, they need to have this transmembrane potential, right? So, and they do this via the use of ATP. They have this sodium potassium ATPase, which pumps sodium out, and is it 3 sodium out and 2 potassium in at the expense of ATP, right? So that's where most of the energy is going, to keep this membrane potential so neurons can fire, and that's why it's prioritized. Here, I just thought it was interesting. Here you see 18-fluorodeoxyglucose we talked about before, PET scan. And we see the brighter the yellow, the more glucose that is being used. You see the brain is very bright, especially in this one region. If we do an MRI, we get a contrast picture. We see, in fact, that there's some kind of diffuse tumor growing in this part of the brain. But the whole brain is dependent on glucose, but the tumor is even more dependent on glucose than the brain. So the tumor will use a lot of energy. Under normal diet, glucose is the primary source of energy, but under starvation, the brain can use ketone bodies, and we discussed a little bit how ketone bodies could be made from amino acids and some fatty acids. So the brain can use beta-hydroxybutyrate, the ketone body from fatty acids, as an energy source for up to 60%. So we still have 40% of its energy needs that will have to come as glucose. The liver uses body fat as a source for making ketone bodies, and then protein to keep the brain supplied. So the liver will start breaking down other tissues in the body to supply the brain with ketone bodies and with glucose, as we'll see when we discuss starvation in the next part of the talk, part 2. So here, I think we're coming to the end of this part. Oh, okay, one more. So where were we with this one? We were with the brain. Okay, yeah. Needs glucose. This glucose typically comes from the liver, but the kidneys can, under starvation conditions, also perform gluconeogenesis to generate glucose to supply it. And then even this figure, better than the other figure I thought, doesn't show the ketone bodies going to the brain. Instead, the ketone bodies are shown going to the heart, but also to the brain, right? And blood, the last organ, and I don't have much to say about it really. It's transport of nutrients, oxygen, carbon dioxide, hormones, and more. Type of cells you're pretty familiar with. Erythrocytes, okay, the red blood cells, a bit unique that they have no nucleus or organelles, and they have a special type of glycolysis to get ATP. They still need energy, right? Despite not having a mitochondria. But you don't need to know much about that. This, okay, important thing here. Blood glucose is between 60 to 90 milligrams per deciliter, or about 4.5 millimolar, and needs to be kept at that. I mentioned it, I'll mention it again. And you can be considered hypoglycemic or low blood sugar at 45 milligrams per deciliter or 2.5 millimolar. And this is the dangerous level of low blood glucose where your brain starts having problems. I think we take a break there. I see how long this is, if I can fit this in here or not.